It's a pleasure to introduce Dr. Vanja Vlajnic. He's a Bayer Science Fellow and Senior Manager within the Statistics and Data Insights function, supporting the late-stage cardiovascular franchise at Bayer. He's the co-lead of the Biostatistics Innovation Center Data Mining Group and is highly involved in developing strategies for the analysis of high-dimensional data in the clinical space. Vanja has a Master's of Applied Statistics from Penn State University, a Master's of Experimental Psychology with a focus in Behavioral Neuroscience from Seton Hall University, and is a PhD candidate in Systems Engineering with a focus on Biomedical Engineering from Colorado State University. He has published in the areas of machine learning applications to clinical data, causal learning and causal inference, clinical assessment tools in schizophrenia, and visual perception. He's a member of the IDS-WG-AI subteam, Phi Beta Kappa, the American Statistical Association, and the Vision Sciences Society. And he's here to talk with us today about wearables and biosensors, applications and analysis in clinical trials. I turn it over to you, Vanja. Thank you.
Yeah, thank you so much. I really appreciate the intro, and thank you again for inviting me to speak here. So I have my presentation recorded, but of course, if anybody has any questions, I'll be happy to answer them in the chat.
Hi, everyone. Thank you for joining me today. My name is Vanja Vlajnic, and I'm a Senior Manager within the Statistics and Data Insights Department at Bayer, as well as a Bayer Science Fellow. And today I want to present some work done in conjunction with some of my other colleagues within the department, more specifically Paolo Piraino and Bogdana Ratic. And the presentation is going to be focused on wearables and biosensors and applications and analysis in clinical trials. So more specifically, what we're looking at is digital biomarker discovery. So essentially, we have several different types of data, whether it's device or device-based subgroups, physical activity features, sensors, and biomarker discovery. And our goal is to assess that through quality of life and disease severity metrics to predict changes over time, to better understand worsening and improvement, and in general, the patient journey and the either the treatment improvement or deterioration over time. And we want to assess clinical outcomes predictors and cofactors, as well as stratification factors, to essentially link digital features to clinical conditions for biomarker discovery and qualification. So just a little introduction about some of the topics that we'll be discussing today. So physical activity is a vital sign, essentially just like blood pressure or temperature, that can be used to monitor and predict health status as well as disease progression, especially in cardiovascular diseases, among others. So the focus of today's talk will be mostly on the cardiovascular space, as this is the area that I specialize in. However, a lot of these topics can essentially be applied to other areas as well. Just because there's different types of digital biomarkers, of course, physical activity only being one of them. So a proper characterization of physical activity by means of single or multiple sensors across the body allows for a more precise classification of patients according to their disabilities, and thus can improve personalized medicine. So physical activity sensors such as wearable device monitors or chest patch monitors, or devices that can be worn on the wrist, for example, can allow for this assessment. Assessing physical activity, especially in normal daily activity in real-life conditions, is extremely complex. It's also very important just because as patients go to the clinic or go to a site to get assessed by a clinician, oftentimes these artificial environments may not be producing what the true ability of the patient is. So, for example, if they're in the clinic and asked to do a 6-minute walking test, the question is always essentially whether or not they're doing more than they feel comfortable with, and we want to get a better understanding of what are the patients capable of on their own time. So what level of physical activity do they feel comfortable engaging in, and what duration do they feel comfortable engaging in on their own. So device vendors, single or multiple pharma companies, and large groups or consortia around the globe are taking different approaches to better propose a valid solution for this. But so far, there's no clinically validated solutions that have been approved that can be used in clinical trials as endpoints. And there's a lot of research, of course, going on in this area. So the present study complements some of these efforts in this space by exploring the clinical utility of a series of features extracted from specifically accelerometer data, which essentially is just a triaxial accelerometer measurement of movement in the X, Y, and Z axes, and these can be used as proxies of physical activity. So as mentioned, I'm going to be focusing on cardiovascular disease and heart failure. So in specific, heart failure is responsible for 11 million physician visits each year and more hospitalizations than all forms of cancer combined. It is important to note that cardiovascular disease and heart failure are often accompanied with comorbidities, including diabetes and renal diseases. More recently, we've seen the implications of the COVID-19 pandemic, especially on how severe of cases patients that have cardiovascular disease or heart failure coupled with COVID can lead to very negative outcomes for patients. So this is another reason why it's important to get a better understanding of cardiovascular disease and try to get some sort of improvement in diagnostic and also treatment opportunities. Furthermore, impacts to quality of life are common in heart failure and cardiovascular disease, particularly in the areas of physical limitation, which is what we'll be talking about today. So as part of the FDA draft guidance on heart failure that was released in June 2019, the agency stated, and I quote, FDA will consider trials that use novel endpoints, including other clinical outcomes assessments, other measures of functional capacity and measures of daily activity, for example, accelerometry data, end quote. But the question is essentially, how do we get from this qualification to actually being able to utilize this type of evidence as confirmatory evidence? So a question is essentially, how do clinical endpoints and outcomes of interest, how do we utilize them to assess the disease state? So essentially on the left-hand side, we have an individual walking, and you can see some sort of a signal based on their activity, intensity, and the duration of their activity. And on the right-hand side, we see an image of a patient wearing a device that can capture this information. Now, how do we take that information and the signal that we're seeing to get a better understanding of the outcome of interest, which is an assessment of the disease state? So what we did here was we had two phase 2b clinical trials, one focused on HFrEF and one focused on HFpEF, with a total of approximately 760 or so patients. And essentially, the trials had a similar design, and there were 4 different time periods throughout the trial where the patients wore devices. So the device was a chest patch that was able to track some of this information on the bottom right-hand side of the screen. So it included heart rate information, min, max, and means, activity intensity, fluid status, respiration rate, posture, and activity duration. On the left-hand side, we see some of the data from non-wearables that was collected as part of the study. This includes left ventricular ejection fraction, NT-proBNP, 6-minute walking test, other clinical blood tests and wet biomarkers, and other PROs. So essentially, the knowledge and challenges that were intended to gain and address here is that we wanted to be able to assess inter- and intra-subject variability from the physical activity data, as well as day-to-day and visit-to-visit variability. Furthermore, we wanted to understand the impact of different data aggregation approaches on clinical interpretation, and to assess advantages and disadvantages of using clinically driven feature extraction approaches as compared to data-driven approaches. Furthermore, the end goal was to essentially build criteria for the selection of the most appropriate and biologically plausible features to use in specific clinical populations out of a series of different types of variants. So essentially what we recognized early on in the work is that not all features would be of importance to all types of indications and clinical populations. So we wanted to hone in on the ones that were most important for the kind of assessments that we were interested in doing. So this is there's a lot going on in this slide, but there's essentially several different types of processes that I want to work you through to get a better understanding of what's involved in the feature extraction process and the feature analysis. So if we look on the left-hand side here, we see the raw triaxial accelerometry data. So this is what the accelerometer is able to essentially record. So you have the XYZ axes that are in units of millig's, which is 1,000th of a gravitational unit. And essentially these are in 3 axes. This is why it's referred to as the triaxial accelerometer. And then from that point, we use that data to then perform QC on the data to ensure consistency and no issues that would indicate problems with the sensor, for example. We assess missing data and missingness in general and perform pre-processing, from which then we extract activity indices, which are the ones that you see below. So there are several different types, since there are several different types more commonly used in the literature. So XL-ACT was one that was provided to us by the manufacturer of the device, and the other ones listed there, ENMO, VMC, square root of the standard deviation, or the square root of the median accelerometry deviations, and the logs of all three of those, or excuse me, all four of those were additionally computed by us as well to see if there's any departures from the daily features that are extracted if one uses different activity indices. So from the activity indice extraction, then we move to the right-hand side where you see the daily activity features that were extracted from this. So as part of this, we extracted several different types of features, including such things as ratios of the total to average activity during the day, the daily activity from 8 a.m. to 8 p.m. divided by the total, the night activity from 10 p.m. to 6 a.m. divided by the total activity, active to sedentary transition probabilities, as well as sedentary to active transition probabilities, as well as other features, including the continuous 6 hours of highest and lowest activities, total activity during the maximum 10 minutes and 10 hours and 14 hours, respectively, of activity, just to get a better understanding of what all these different types of activity features would provide us. And then specifically, we performed comparisons, including density distributions, correlations, pairwise scatter, multivariate analysis, and changes by day and per visit to get a better understanding of what's happening from an interpatient and an intrapatient variability perspective over time. So now we can transition over and take a deeper dive into that data. So essentially, this is what the data looks like when it comes from the device. So you have essentially a timestamp and the XYZ axes, and this is then being tracked for the patient essentially throughout the course of them wearing the device. So this specific device, as you can see from the timestamp, you have a tracking of every 4 seconds. For every 4 seconds, you have additional XYZ axes being tracked. There's some devices that can track data on a higher resolution and a lower resolution, but this was the specific resolution that we had with it. So this slide just speaks to some of the data pre-processing and aggregation steps. So as I mentioned, we have 4-second intervals in the data. So if you have 24 hours in a day, 60 minutes in an hour, 60 seconds in a minute, and you have 4-second intervals, you end up with 21,600 observations per day for 100% compliance. So as you see on the right-hand side, this day the patient had 20,610, so almost the full 21,600 observations. For some of the days, you had the full 21,600. So the first day, most likely the patient was wearing the device or put on the device, let's say in the middle of the day, so they missed approximately half of the day's activity data. That's why you only see 12,000 approximately observations for that day. Other times you can see things like this, such as what's shown in this graph, where essentially you have a large gap at a certain portion of the day. Perhaps the patient took the device off in the middle of the night and put it on later in the day. So these kinds of data were excluded from this analysis. Later, we performed sensitivity analyses to understand the missingness aspect a little better. But for this initial assessment, these missing data type days where we had a lot of data missing were excluded. So with respect to the activity indices, we selected the appropriate options as shown below. So several activity indice derivations are available. As mentioned, XL-ACT was the one that was proprietary from the manufacturer, and essentially we wanted to use some of the open source ones that are listed here to get a better understanding essentially of how well does the activity index that was provided from the manufacturer match up with the ones that are in the open source literature, and essentially how well would we be able to reproduce this feature extraction process, or would we essentially end up with different conclusions based on the different indices that we could be using. So as mentioned, we have the Euclidean norm -1. the median accelerometry deviations, the square root of standard deviations, and the vector magnitude count approach. With respect to activity indices, so what we wanted to get a better understanding of was how to address the multi-device and multi-manufacturer problem across time. So it's clear that we're going to end up utilizing wearables and sensors throughout the future course of clinical trials, as it can supplement our information about the patients and just their daily habits and activities that they're able to engage in. So activity indices are important because they allow us to go from the triaxial accelerometry data, essentially the raw data that the sensor is capable of tracking, to a measurement of activity, which is the outcome of interest that we're interested in. So we need this data and a pipeline to compare across trials and therapies where appropriate, and thus we also need it to be device agnostic, because it's clear that it's not going to work if we have different devices that are being utilized in the course of the research and development process. However, if each device is using a different way to produce these activity indices and also the features that we're interested in, we might end up in a case where these features aren't comparable, and there's not going to be a way to assess features derived from one device as compared to features derived from a different device, even if it's within the same population. So as manufacturer-derived activity data is often a black box, in a black box format, essentially to protect the IP of the manufacturer, we wanted to also look at some of these other indices that I have mentioned previously. As you can see on the right-hand side, these are some of the other indices plotted as compared to XL-ACT, which is the one on the bottom. And essentially, you can see different signals from the same activity data, and we wanted to get a better understanding of, okay, how does this relate then to our ability to understand the data that comes from these activity indices? So initially, to get a better understanding of the data that we were looking at, we wanted to assess the smooth data. So essentially, to take the signal that we had from the activity intensity, the activity indices, and essentially smooth them over time. So we tried 12 degrees of freedom for smoothing, and then we also tried 24 degrees of freedom for smoothing, essentially associated with 1 degree of freedom per hourly day. So essentially from 12 a.m. to 12 p.m. for the 12 degrees of freedom. And then we also wanted to try one where there's 1 degree of freedom essentially for every hour within the day, so 24 total. And we looked at how the essentially how the patient profiles differ when looking at the different smooth activity, and essentially whether the 12 degrees of freedom would provide overly smooth data, or if we would still be able to see the signal and the nuances between the two. So this is an example for one of the sets of patients. You can see that sometimes there's missing data. So for example, for this the second patient here, the end of treatment data is missing because the patient most likely never attended that visit. But you can see in general what you would expect to see in most activity data. So you can see that there's a spike throughout the day. Patients are engaged in some level of activity, and then kind of tapers off towards the evening, of course, when it goes into a resting period. So for some patients, you can see that this pattern carries over throughout the course of the trial. But then for other patients, you can see that even in the beginning, they don't have a lot of, or at least they don't engage in a lot of activity. So this patient pretty much stays flat across the course of the trial. This one's interesting because they start off relatively flat on their activity, and then there's a spike at the second visit, and then essentially it tapers down over the course of time. So these are the kinds of things that you can kind of holistically get an idea of, especially if you take this data and superimpose it on, or take the average across all the different patients. Other things that we looked at were assessments of distribution and Spearman correlations for the different indices. So on the left-hand side here, we have density distributions. As we can see, a lot of this data isn't normally distributed. So what we also did was take the log of the data to get a better understanding of what it would look like. It makes sense that it's not going to be normally distributed as well, just because a lot of time is going to be spent not engaged in a lot of activity. There might be initial spikes throughout the day, but it's clear that the data isn't going to be normally distributed, and some form of transformation is going to be necessary. We also wanted to assess how well the different indices correlated with one another, and you can see that there's a high level of correlation between the square root standard deviation and square root MAD approaches and also the vector magnitude approach and the NMO approaches. And you can see this also, it makes sense if you look at the slide referenced earlier for their computations. The two that correlate with one another, the two sets that correlate with one another have very similar computations as well. So essentially what we see on this slide is the daily features computed across a random subsample of our patients. So on the x-axis, you have the 4 different time periods throughout the course of the trial that the patient was wearing the device. And essentially on the y-axis, we have different activity features that were computed. So we have the first one being the active to sedentary transition probabilities, the other ones here being different measures of either daily activity, night activity, measures of total activity and continuous 10 hours, 14 hours, so on and so forth. So what's nice about looking at the data in this way is that one can essentially get an idea of how the activity transitions over time throughout the course of the trial. So as mentioned, these are computed across a series of patients, a random subsample of the patients that we had in the trial. And from that, one can look at and get a better understanding of perhaps here, for example, you see that there's this is the mean activity throughout the day. So it starts with a spike in activity during the day, and then it decreases, and then it increases again over time. You can see that there's very cyclical natures within this data, and also it's interesting to look at across time for certain variables. The pattern doesn't change very much. So over here we can see that we have different patients here presented. Some of them are continue to have high activity throughout the day. Some of them have lower activity, and you can essentially see that in some cases this changes over time. In other cases, it remains relatively constant. And what's nice here is that one can utilize this kind of approach to look at outliers in the data, or just to, in general, get an idea of the differences. So you obviously have patients that are less active, patients that are more active, and essentially look into that further within the data. In addition, we wanted to look at the correlations within and across patients. So on the right-hand side over here, you can see again a random selection of patients, and we can see their correlation structure amongst different features. You can see certain patterns arise that the different total activity measures are relatively well correlated and highly correlated amongst all of the patients. There are certain variables that are less correlated, but in general, there seems to be a pattern arising here. When we look at across the patients, again, we see that this selection of total activity, whether it's 10 or 14 hours or 10 minutes, there's high correlation in the total activity of the highest level of activity across these areas. There's other variables that have a low correlation between themselves and other aspects of the feature extraction as well. There's some that are not correlated whatsoever. And it's also just interesting to note the different relationships among some of the features that are extracted. So what we notice here is that essentially we wanted to look at whether features perform similarly or differently. And an example here is we have the DARE day, which is defined as the total activity during 8 a.m. to 8 p.m. divided by total activity. And then we also have total activity in the continuous 10 hours, which is essentially defined as the max value of total activity, 10-hour windows shifted every 4 seconds. So in these plots here, we have preserved and reduced ejection fraction, and this is essentially the categorization of heart failure patients, whereas if their hearts are able to pump out blood sufficiently, then they fall within the preserved ejection fraction category because their heart, their ability to eject blood essentially out of their heart is preserved, whereas if this is compromised, then they would fall into the reduced ejection fraction category. And then we also have summary statistics, including the mean and the median. So if we look at DARE day, for example, we see in the preserved group that there isn't very much of a difference across time. And when we perform the Kruskal-Wallis test, the p-value is not significant. And also if we look at the comparisons, they're not significant over time. What's interesting is that when you look into the reduced group, some of the comparisons are different across time. So you see that the changes over time from earlier parts of the trial to later parts of the trial, you're able to detect some of these differences, which is particularly interesting. And then the opposite happens over here for total activity of continuous 10 hours, where in the reduced group, we see that there's not much of a difference when comparing earlier parts of the trial to later stages. Whereas if you look at, for the preserved group, once you compare earlier parts of the trial to later parts of the trial, you have more indication that there are significant changes occurring. So comparisons of two features extracted from the same index and the ability to detect change across visits for two different populations. This essentially highlights the fact that one can have different features that are more appropriate for specific populations to be able to assess changes over time. And this is the kind of work that we wanted to look into to get a better understanding of what those features are and to be able to assess them from a validation perspective in some of our other trials moving forward. So some of the outcomes of the project in general, so the inter and intra-subject variability as well as day-to-day and visit-to-visit variability suggest that each parameter performs differently and thus necessitates an appropriate selection. So as mentioned earlier, it's very important to be able to get a better understanding of the clinical outcome of interest as part of the study that one is looking to perform. And it's clear that different types of features are going to be important for different types of studies and different types of populations of interest. So in order to be able to track and understand one certain aspect or one clinical symptomology across time, there may be features that are more implicated in this, and there's more appropriate features to be selected. So the impact of data aggregation on clinical interpretation is not neglectable and needs to be considered when working with high-resolution data collected in different time periods. An understanding of the advantageous and disadvantageous aspects of using clinical-driven feature extraction approaches as compared to data-driven approaches was developed. So evidently there's, of course, a lot that can be gained by using clinically driven feature extraction as well as data-driven feature extraction. And together with clinicians and other medical device experts, the team needs to come together to really decide which feature extraction process is most appropriate and which features derived from either a data-driven approach or a clinically driven approach are most appropriate to utilize in the assessment of these patients and the changes over time. And in general, we gained a knowledge to build criteria for the selection of the most appropriate biologically plausible feature to use in a specific clinical population. Some of the references for some of the work that we. So thank you. That concludes my presentation. Please feel free to reach out to me either on LinkedIn or directly here, leaving me a message or within the chats, and I would be happy to take any questions now. Thank you.
Yes, thank you. So thank you everyone for listening to my presentation. I've tried to keep up with some of the questions coming in. I don't know if there's any other additional questions that anybody has that may want to put them into chat, or of course, feel free to contact me on LinkedIn after the presentation.
Thanks, Vanja. That was a very interesting presentation. I think I had really just one question based on the conclusions that you came to after the study and the analysis, and that's really, do you plan to add more patients or more data, continue adding data to this data set in order to be able to better understand, some of the selections that you might want to make or focus more on as a result, or are you essentially finished with this particular study?
Yeah, that's a good question. I mean, these were the utilization of these biomarkers and wearables were particular to one specific study, or two studies actually that we were running. But we have additional studies that we're running different kinds of wearable devices in, accelerometers, for example, and as well as for different therapeutic areas, we're also looking into some other devices to utilize. So in general, I would say that the pharmaceutical industry is moving towards the utilization of digital devices a lot more in clinical trials, particularly as it helps for gaining information about the patients, not only from a regular patient journey perspective, but you're also getting supplemental information that you might not have already been able to attract attention to as part of the regular clinical trial setup. And it also allows for the possibility of patients that might not be able to be involved in the trial in the first place. So if they're located too far away from where the trial is being set up. So yes, to to summarize, essentially, Bayer is definitely doing a lot of work within the digital device landscape, and I know a lot of other colleagues at other sponsor companies as well.
Can I add a follow-up question?
Sorry. Sure, of course.
We get to the one in the chat. You did show at least a couple of instances of, what you thought were people removing the device maybe before going to sleep. Were they given guidance at the beginning of the study to try and keep the devices on 24/7, and that just didn't always follow through in practice? Or can you comment on that?
So there's exactly. So there was some guidance that they were given as part of the study in the beginning. So essentially, this was one device that was worn on the chest, and there was essentially some sort of sticky material to ensure connectivity between the device and the chest. So if the patients were going to take it off, then they would have to reapply the material to ensure that it sticks appropriately, and it added layers of complexity to just the entire process. But these are the kinds of things that you're not really aware of until it actually run them in a clinical trial or in a study to get a better idea of what the issues may be. Some things that we've discussed is perhaps if you're just interested in actigraphy, in that case, it's easy just to give a patient some sort of a device on the wrist, like a watch, for example. You can have, that makes it easier because then it's simpler for them to take it off and put it back on without having to worry about where exactly they're placing it on the chest. It becomes more complicated to get measures of fluid retention and heart rate, just because obviously it's not as precise on the wrist. There's also some devices that are sort of one-time use. So essentially the patient can put them on, and if they feel like they can't go through, let's say, a week at a time of wearing the device, then they just take the device off and then they would dispose of it. So there's difficulties therein, and we're also trying to assess whether some of the missingness is informative. So it might be the case that let's say hypothetically a patient is wearing a device and then starts not feeling well, and then they decide to take the device off. So if there's a lot of patients that stop taking the device prior to some sort of a deterioration, then that would be informative, and that might be a pattern that we would want to be paying closer attention to, essentially. But yeah, great. Thank you. That was a great question. Perhaps I can get to this one in the chat. So the question is, was this data taken during the pandemic? How do you take into account large perturbations like season, weather, or pandemic into this data? So that's a great question. So for this data specifically, it wasn't taken during the pandemic. So this was prior to COVID, luckily. Although on the flip side, I think that particularly with the pandemic, we've seen how, obviously it's had a lot of impact on the pharmaceutical industry and in clinical trials as well, with patients not being able to attend site visits. So perhaps having devices like this where the patient's in their home or doing whatever they do from a daily activity perspective, then you would still be able to be getting some information about the patient without them having to attend a visit. There's definitely impacts to seasonality and weather that we've noticed within the data. So what we try to do is also pull in information about the weather, or at least the season that the patient is currently in, just because you can imagine that if someone is experiencing or living in an area where it's currently summer, they might be more comfortable walking around more or engaging in physical activity, versus if you're in some area that's currently experiencing very cold weather, you might not be going outside very frequently. So that can obviously have an impact on people's physical activity. So these are definitely things that have to be taken into account. There's other aspects that we've noticed, such as weekday versus weekend effects. So oftentimes during the weekday, people usually follow the generally it's the same kind of setup throughout the week, but then physical activity often seems to change on the weekends compared to the weekdays. So these are other things that we try to take into account as an example. Thank you for the question.
Thank you, Vanja. Sam, did you have any last comments?
Oh, so from my side, no. Thank you, everyone, for listening to the talk. And as I mentioned, if anybody has any questions, please feel free to follow up with me. I think that in general, the wearable and biosensor space is going to continue expanding over the next couple of years. And I think that there's a lot of use cases from cardiovascular disease to neurological diseases like Parkinson's, for example, also to There's some work being done, I know, in the cough sector, because then you can use the device to measure essentially whether the patient's coughing or the frequency of coughs. So there's a lot of utility in this, and I think that it has the opportunity to really help supplement a lot of the information and understanding of some of these disease states. So thank you again for having me and listening to my talk, and it's much appreciated.
Thank you, Vanja.
Thank you. Bye-bye.